This is Radio Health Journal. I'm Nancy Benson. This week, have scientists finally found the key to healing an injured brain or spinal cord?
When we inject these human cells into mice, it stimulates nerve fiber growth and also rescues dying nerve cells.
Hope for spinal cord injuries, and maybe even MS and ALS, when Radio Health Journal returns.
I'm Reed Pence, the producer and host of Radio Health Journal. If you like listening to Radio Health Journal, you'll also like our sister show, Viewpoints, which covers a wide array of topics from education to history to the environment. Here's a preview of what they're covering this week on Viewpoints.
He's often drawn as half genius, half jerk from birth. That's not accurate.
The lasting legacy of the late Steve Jobs. Then,
most of us revert to some sort of what we call an adapter. It's a little nervous gesture that we use, a self-soothing gesture like rubbing your fingertips together.
What signs do you show when you're feeling tense or telling a lie? I'm Marty Peterson.
And I'm Gary Price. These stories in depth this week on your public affairs magazine, Viewpoints.
Listen to Radio Health Journal and Viewpoints on your favorite radio station. And subscribe and listen anytime on Apple Podcasts, Google Play, and Spotify. Also, follow us on Instagram and Twitter at Radio Health Journal.
About 80,000 Americans every year suffer some kind of permanent disability as the result of a traumatic brain injury. Another 17,000 suffer a spinal cord injury causing some form of paralysis. Recovery of function after such an injury is rare because, unlike bone or muscle cells which can heal themselves, central nervous system cells cannot.
In the adult nervous system, nerve cells do not divide. So once they die, they're not replaced. Furthermore, nerve fibers, once they're severed within the central nervous system, do not regrow.
That's Dr. Benjamin Segal, chair of neurology and director of the Neuroscience Research Institute at The Ohio State University.
There are some natural obstacles for regrowth. Some of these are different molecular signals that prevent nerve fiber regeneration. Sometimes there's also scar tissue that interferes, but it's been a big challenge in terms of neurological therapeutics to overcome. The peripheral nervous system outside of the brain and the spinal cord has much greater capacity for nerve fiber regeneration. But within the central nervous system, there are all of these roadblocks.
However, there's finally a little optimism that some of those roadblocks can eventually be overcome. In a study published in the journal Nature Immunology, Segal's team has uncovered the existence of cells in mice that can signal nerve cells to repair themselves.
This is a white blood cell. It falls into the general category of white blood cells called granulocytes that contain granules with soluble factors that they can release. It's in an immature state of development, but it is unusual. This particular cell type hasn't been described before based on the genes it expresses and the markers it expresses on its surface. And what's really unusual is that it produces a lot of growth factors that promote the health of nerve cells and stimulate nerve fiber regeneration. We also have been able to isolate these cells and inject them into mice with central nervous system injury at the site of injury, and they are protective and promote repair.
Some of the repairs these cells have triggered have never been seen before.
We find that if we crush the optic nerve with forceps, normally the nerve fibers don't regrow and the nerve cells connected to those fibers die. When we introduce these cells, this immature granulocyte, we see a dramatic rescue of the nerve cells from death, and also they begin growing nerve fibers, which, is very unusual, unprecedented. We then wanted to know, is this specific for the optic nerve, or can it occur in other compartments of the nervous system? So we did similar experiments in the spinal cord. So if we injure the spinal cord and introduce these immune cells, these special immune cells, it also stimulates nerve regrowth in the spinal cord.
Segal says his initial tests applied the cells to the injury site immediately after the injury, but he realized that growth factors would have to work for some time after an injury, perhaps a few hours, to be useful in the real world.
So we did some experiments where we delayed the transfer of these cells following the injury, and we found that after a significant delay, we could still transfer these cells for therapeutic benefit, and that they still were able to rescue the dying nerve cells and stimulate new nerve growth. There was a limit, though. If we waited too long, then they became less effective. But definitely there was a substantive time window when they were still able to function in a therapeutic manner.
Segal says the window of treatment appears to be about 12 hours in mice, though it may be different in humans. But just how much healing can the granulocytes accomplish? Is a crushed optic nerve healed enough to allow mice to see again? Researchers don't know yet.
That is next on our list. We didn't wait long enough for the fibers to grow to the extent that they would be able to complete their circuits. We have done some experiments where we've waited longer and shown that in the optic nerve, for example, the nerves will grow all the way back to the brain. But now we're going to do functional studies to see if there's any evidence of restoration of vision, and in the spinal cord, whether there's any improvement in terms of sensation. Because in the spinal cord, we severed nerve fibers involved in sensation.
The results so far have produced optimism, but it's a long way from mice to people. And while the granulocytes occur naturally in mice, they've been a little tougher to track down in humans.
What we did find is a human cell line which has characteristics of an immature granulocyte, and we showed that this human cell exhibits similar properties to the mouse cell in that it does release these growth factors, and when we inject these human cells into mice, it stimulates nerve fiber growth and also rescues dying nerve cells in mice, and it also works in a petri dish with explanted nerve cells. So if we take human nerve cells in a dish and add this human cell line that resembles the mouse pro-regenerative granulocyte, it stimulates those nerve cells to grow new nerve fibers. Now we're looking to discover or identify the naturally occurring counterpart in humans.
If those efforts are successful, Segal says growth factors could be applied to far more than just injury. In theory, they could help repair damage from degenerative diseases like ALS and MS.
In diseases like multiple sclerosis, for example, we know now that there's actually loss of nerve cells. There's also transections of nerve fibers. So nerve fibers actually get split in half at the site of MS lesions or MS plaques. Theoretically, these immune cells may be able to decrease cell death in MS and stimulate some of those severed fibers to regrow across plaques. ALS involves the dying and the retraction of nerve fibers in the spinal cord, the nerve fibers that innervate muscles. So theoretically, it's possible that these cells would be protective under those instances.
Segal says visual diseases like glaucoma could also be helped if nerve fibers can be repaired. And his lab is starting to investigate animal models to repair the damage of stroke. If granulocytes can prompt nerve repair almost anywhere, the possibilities are nearly endless. You can find out more about all our guests on our website, radiohealthjournal.org. Our studio producer is Jason Dickey. I'm Nancy Benson.
Radio Health Journal returns in just a moment.
With many opportunities to celebrate during fall and winter, preparing foods for all the festive occasions can be both a joy and a challenge. Selecting basic foods that can work in multiple ways, such as fresh grapes, can simplify things greatly. According to registered dietitian Courtney Romano, health advisor to the California Table Grape Commission.
Fresh grapes are beautiful. They taste great and are easy to keep around for everyday eating as well as special occasions. Keep some on hand for a healthy snack option. Arrange them in a pretty bowl for a classic and edible centerpiece, or in small clusters on a serving platter. Use them as a special ingredient. Heart-healthy grapes can be dipped in dark chocolate for a delightful dessert that is good for you too.
Heart-healthy grapes from California are in season from May through January. Grapes of all colors, red, green, and black, are a natural source of beneficial antioxidants and other polyphenols, which may help contribute to heart health. Visit grapesfromcalifornia.com for more information.
Holidays are a time to talk to family members about heart health. Encourage them to see their doctor if something doesn't feel right. For example, aortic stenosis, one of the most serious and common forms of heart valve disease, especially affects people over age 65. But Dr. Vuisil Nkomo of the Mayo Clinic says people who have AS may not get the medical attention they need.
Symptoms of aortic stenosis may include chest pain, significant fatigue, shortness of breath, and difficulty walking short distances. People often dismiss these symptoms as simply a part of aging. AS can be treated, but if it is not, it can ultimately lead to heart failure and death.
If you have symptoms, see your healthcare professional for a heart function and valve check. Target Aortic Stenosis from the American Heart Association, sponsored by Edwards Life Sciences, has tools for patients and families at heart.org/AS. That's heart.org/AS.
And that's Radio Health Journal for this week. Radio Health Journal is a production of MediaTrax Communications. Follow us on Twitter, Facebook, and Instagram to learn more. And check Apple Podcasts, Google Play, and Spotify for a library of past programs. Plus, you'll always find previous segments and information about our guests at radiohealthjournal.org. Join us again next week for another edition of Radio Health Journal.
Coming up next week on Radio Health Journal.
I've seen those numbers, and I'm afraid they are not alarmist. They are quite realistic.
COVID numbers are skyrocketing. Is it possible deaths could climb to 500,000 by February 1st?
Then hiccups. Almost everybody has a favorite folk remedy to try to get rid of them. They may not be based in science, but most of those remedies actually work.
I think they work because they're forcing you to contract your diaphragm muscle, to twist your ribs, to wince when you're eating something like lemon and sugar.
All that and more on Radio Health Journal.